Hello guys, welcome to this lesson. We're going to talk about some studies related to primary structures of proteins. But before starting, I want to tell you what we're about to learn. We will talk about what are amino acid analysis, how Edmonds degradation work, and what is protein fragmentation. So let's get started. Amino acid analysis is a laboratory technique used to determine the composition and concentration of amino acids in a protein or peptide sample. This information is crucial for understanding the structure and function of proteins, as well as for various applications in biochemistry, nutrition, and clinical research. However, this technique does not inform the order of amino acids in peptides and proteins. In fact, it requires another study that we will shortly cover. Overall, amino acid analysis is a powerful tool for understanding the molecular makeup of proteins and peptides, providing valuable insights into their structure and function. Let's see how amino acid analysis works. The first step involves preparing the protein or peptide sample for analysis. This often includes hydrolyzing the protein and breaking down the peptide bonds between amino acids. Acid hydrolysis or enzymatic digestion can be employed depending on the specific requirements of the analysis. As the peptide bond is quite strong, the process may require 24 hours in order to break down the bond. The amino acids in the hydrolyzed sample are often derivatized to make them more amenable to detection. Derivatization is like giving a makeover to molecules. Imagine you have a group of molecules and you want to make them more visible or easier to work with in a laboratory. Derivatization is a process of adding a kind of tag or special feature to these molecules, making them stand out and be more detectable. This tagging or modification helps scientists study and measure the molecules more effectively. In simple terms, it's like putting colorful stickers on things you want to find easily in a busy room. Derivatization helps scientists see and understand molecules better, making their characteristics more apparent and easier to analyze. The derivatized amino acids are then separated based on their chemical properties. High-performance liquid chromatography, also known as HPLC, is a commonly used technique for this purpose. In a nutshell, HPLC is a method to sort and identify different substances in a liquid mixture, providing valuable information about their presence and quantity. The separation is typically achieved using a chromatographic column with specific properties that allow amino acids to be eluted at different times. I know all these terms may be confusing, so let's take the time to understand what a chromatographic column is and how it works. Imagine a chromatographic column as a tiny maze for molecules. Molecules are injected into one end of the column like racers getting ready at the starting line. Inside the column, there are tiny particles acting like obstacles in a maze. Each molecule has its own way of navigating through this maze. Some molecules move through the maze quickly because they don't interact much with its obstacles. Others might take longer because they interact more. As molecules exit the maze, they reach the finish line. Here, a detector sees them, and it's like capturing the racers finishing their race. Each molecule has a unique speed, so they finish the race at different times. This separation helps identify and distinguish them from one another. Coming back to the amino acid analysis, once separated, the amino acids are detected. Detection methods include UV absorption, fluorescence, or mass spectrometry. depending on the specific derivatization used. The choice of detection method often depends on the sensitivity required and the equipment available in the laboratory. The amount of each amino acid is quantified based on the intensity of the detected signal. The final step involves analyzing the data obtained from the amino acid analysis. Software is often used to process chromatograms and calculate the concentrations of individual amino acids in the sample. The resulting amino acid profile provides information about the protein's composition. Amino acid analysis finds diverse applications across various domains. Primarily, it plays a fundamental role in protein characterization, aiding in the determination of the primary structure of proteins. In the food and pharmaceutical industries, this analytical technique is crucial for quality control measures, ensuring the uniformity and high quality of products. Additionally, amino acid analysis is instrumental in nutritional analysis, providing valuable insights into the amino acid content of food items and dietary supplements. In the realm of clinical research, the technique is employed to study amino acid imbalances or abnormalities in patients, contributing to a deeper understanding of health conditions. Furthermore, amino acid analysis is an essential tool for protein quantification, often used in combination with other techniques to achieve accurate and reliable measurements of protein concentrations. Overall, the versatility of amino acid analysis makes it an indispensable tool in diverse scientific and industrial applications. Now let's talk about Edmonds degradation. It is a chemical method used for the step-by-step sequencing of amino acids in a peptide or protein. It allows researchers to determine the primary structure of a protein by identifying the order of amino acids linked together in the polypeptide chain. Let's see how Edmonds degradation works. Edmonds degradation relies on the selective removal of the N-terminal amino acid from a peptide or protein without affecting the rest of the molecule. The removed amino acid is then identified and the process is repeated to sequence the entire polypeptide chain. The procedure begins with the reaction of the N-terminal amino group with a reagent called phenylisotiacyanate, PITC. This forms a cyclic structure derivative of the N-terminal amino acid. The next step involves cleaving the N-terminal cyclic amino acid, releasing the labeled amino acid without affecting the rest of the protein. The released amino acid can then be identified by various analytical techniques such as chromatography or mass spectrometry. The derivatization facilitates easy detection. The remaining polypeptide chain now lacks the original N-terminal amino acid. The process is repeated on the shortened chain to identify the next amino acid in the sequence. Edmonds degradation is an iterative process. Each cycle removes and identifies one amino acid at a time, gradually sequencing the entire polypeptide chain from the N-terminus to the C-terminus. However, while Edmonds degradation is effective for short to medium-sized peptides, it becomes less efficient for larger proteins due to cumulative errors in each cycle. Modern sequencing methods such as mass spectrometry and automated Edmonds sequencing machines have largely replaced manual Edmonds degradation for large-scale protein sequencing. Now, let's focus on protein fragmentation. Protein fragmentation refers to the process of breaking down a protein into smaller peptide fragments. This fragmentation is often done in order to analyze proteins or polypeptides. Two common methods for protein fragmentation are enzymatic digestion and chemical cleavage. Let's see part of the enzymatic digestion. We find trypsin digestion. Trypsin is a proteolytic enzyme that specifically cleaves peptide bonds on the carboxyl side of the lysine and arginine residues, as you see over here. When a protein is treated with trypsin, it cleaves the protein at these specific amino acids, resulting in a set of smaller peptides. Then we also have the example of chymotrypsin digestion. Chymotrypsin is another proteolytic enzyme, but it cleaves at the carboxyl side of large hydrophobic amino acids like phenylalanine, tryptophan, and tyrosine. Then we also have chemical cleavage, with the example of cyanogen bromide, CNBr. It is a chemical reagent that cleaves proteins at the carboxyl side of methionine residues. This method is useful for proteins containing multiple methionine residues. Fragmentation plays a pivotal role in diverse applications within the field of protein analysis, primarily in a mass spectrometry-based proteomics. Fragmentation is a key tool for protein identification in complex mixtures, enabling researchers to discern the presence and identity of proteins. Additionally, fragmentation patterns are instrumental in structural studies, offering valuable insights into the three-dimensional architecture of proteins. In the realm of sequencing, particularly in de novo sequencing or when dealing with unknown proteins, understanding fragmentation patterns is essential for accurately determining the sequential arrangement of amino acids in proteins. These applications collectively showcase the significance of protein fragmentation in unraveling the intricacies of protein composition, structure, and sequencing for various scientific investigations. Well guys, this is all for this lesson. I hope you like it, and I'll see you in the next one. Bye bye.